My name is Phil Ewels, and I work at this SciLifeLab, Science for Life laboratory here in Sweden. SciLifeLab is a collaboration between the major universities in Stockholm and Uppsala, and we're basically a focus for molecular biosciences, so anything to do with biology research in Sweden. SciLifeLab has lots of different research groups within it, and we also have these different platforms and facilities that offer technical services to research groups. So we can centralize kind of knowledge and infrastructure in a single place and make that available to researchers all across Sweden and beyond. And so I work at one of these technology platforms, which is called the National Genomics Infrastructure. And as the name implies, we work with genomics, which is basically DNA sequencing and everything involved around that. We primarily do two main things. We offer guidelines and support for people, so people who are new to this relatively new field of DNA sequencing can come to us and ask for help. We offer the infrastructure and the technical know-how to actually help them with their hands-on experiments, and we're focused in Sweden. On a day-to-day basis, we do lots and lots of user meetings with people. We do the actual library preparation. I'll come on to a bit more in a second about what this means. We do the DNA sequencing, actually figuring everything out, and then we finish up with the data analysis and delivery, which is where you guys are probably going to be most interested. Okay, so in order for everything else to make sense, I'm going to start off with like a little bit of a primer as to what genomics is, how DNA sequencing works, and what all this stuff is that I'm talking about. So, this is me, if you like, typical person. I'm made up of mostly cells. Hopefully everyone's done enough biology to be familiar with this concept. Cells are really, really small, and there are lots of them that make me up. Estimates vary, but probably about 30 to 40 trillion cells in my body. If you include bacteria, that number goes up to like 70 trillion. But just my cells, about 30 to 40 million. That's 30 million cells. It's a big number. Each cell itself is very small, again in range here, but say about 25 micrometers, so that's 25 millionths of a meter. And within each one of those cells is this little blob in the middle called a nucleus, which is about 6 micrometers. Inside that nucleus, we have our DNA, which is this familiar double helix structure. It's a biomolecule, and it encodes all of the information pretty much needed for life. It describes how to make me. In one of those little cells, I have my genome, and if I stretched all of that DNA out into one really, really, really long line, which would be impossible to do, but just bear with me. If I stretched it all out, it'd be about 2 meters of DNA in a single cell. And remember, this cell is very tiny, so that's a lot of DNA. It's very, very well packed, very compressed. And for this talk, for the first time, I actually did the calculation and was like, okay, if I took all the DNA out of my body, it's about 600 grams of DNA, I think, and I put it in one single line, how far would it go? I was like, okay, 80 billion kilometers, that sounds like a lot. What does that, what's something, in terms I can understand, what is that? And it's about the diameter of the solar system, times 2. So there's lots of DNA in us, which is a good thing. This might be totally overwhelming. Like, how can we possibly do anything with this volume of information? But what we're saved by in genomics is that each cell contains one copy of my genome, and that genome is the same in every cell. So that massive amount of DNA is basically 40 trillion copies of the same thing. So, what's DNA made up of? Everyone's seen this stylistic kind of double helix structure. What is it? DNA is made up as a polymer, made up of 4 base units, which are called bases. There's adenine, cytosine, thymine, and guanine, which we simplify to the 4 nucleotide letters. And they stick together. So A always binds to T, and C always binds to G, and that gives us this double helix. So the double helix is made up of 2 pairs of these things stuck together in a sequence. And it's this sequence which is interesting, and this helps us whenever we do data science, because we can just represent one of those strands and see the order of letters. It's a bit like kind of binary code. The zeros and ones aren't interesting by themselves, but it's the context, it's the sequence of those numbers, or in this case, those 4 letters, which encodes the information. And when the Human Genome Project was finished, there was an art installation, which you can go and see in London, where the human genome was being printed out in books, and there's a lot of it. It's a whole bookshelf, but probably not a very interesting read. But for your computer, it's really good. So, final bit of introduction. How do we go from a sample, which a researcher has generated, could be a blood sample, could be from an animal in the wild, like a sea slug, or name your favourite creature. How do we go from something like that to something we can process, some kind of data analysis? We make use of the same trick that biology uses with DNA, which is that these magic sticking pairs mean that we can unzip that double helix of DNA into 2 separate strands. We can get rid of one of them, and then we can make a copy, one base pair at a time. So we stick on the starter linker, and then we use the 4 bases, but this time we put in fluorescently labeled bases, and each one of them is a different color. We add them one at a time, and then we use a microscope to visualize that pattern across basically a microscope slide. And so what this picture shows, each one of those dots is a cluster of DNA sequences, which are the same, and each color indicates what that base pair was at that point. And so as you do this reaction, stepwise, you do it 50 times, 100 times, you get those 100 bases across each one of those very many sequences. So you don't do very much DNA sequencing, but you get it's very, very highly parallelized, and you end up with lots of data. So that's what we're working with. That was quite abstract, so I thought I'd put some photos in to show you what it actually looks like. So we start off by sending these out to research groups. These little wells just to contain samples. There's 96 of each plate. They send back some DNA. This is Anna in my lab, and she's prepping up that DNA and basically doing a bunch of chemical reactions to pre-process those samples into something we can work with. We stick it in various machines and kind of spin it around and put it in tubes and things. And when it's ready, we stick it on one of these. This is a microscope slide that I talked about, which we use for DNA sequencing. We also have these, slightly different technology for doing a similar thing, called about this big. One of these has gone up to the International Space Station just recently to sequence DNA up in space. This is a different type, and this is a different type. And once the DNA is loaded on that microscope slide, we can stick it on a machine and do that experiment. It looks like this. At the back of the machine, we have this, and this is where we're all a bit more familiar. Lots of stuff goes through these and ends up on one of these. And this is where data science comes in. Cool. It wouldn't be a genomics talk if I didn't put up this slide. Jared introduced Moore's law in computing, and this is the genomics equivalent showing how much it costs to sequence a single human genome over as years have progressed. And you can see we're outpacing Moore's law by a long way. This is a log scale on the y-axis. And so just in the time that I've been studying genomics, the price and therefore the volume at which we can sequence DNA has dropped just fantastically, which really has opened up a whole new era, a whole new branch of science. And now we can do things which were completely unthinkable only a few years ago. To put this into context, the first human genome took about 13 years to sequence. It was a massive collaboration across labs all over the world. Back in 2016 at NGI Stockholm, we did one of these human genome equivalents every 4 minutes, throughout 24 hours a day throughout the whole year. It's a massive change. In that year, we delivered something in the order of about 200 terabytes of processed data to our research groups. And this is the finished, compressed, just the useful stuff. The intermediate data is much, much bigger than this. And in order to do this, we have a compute cluster, traditional kind of offsite compute cluster with about 4,000 cores, a lot of RAM, 1 to 2 petabytes of disk, which has been updated. Okay, that was my brief introduction finished. Hopefully I've got across that the field of genomics generates lots of data and it's changing very, very fast. New things are becoming possible all the time, which opens up new avenues of research. So when it comes to actually doing the data analysis, once we've got all these sequence letters in order, we've got this massive amount of data, how do we actually do something interesting with that? So that human genome, reference genome, is about 3.2, 3.5 billion base pairs of sequence in order. And now we're looking at data, not DNA. So instead of that double helix, now we've just got a string of letters. And when we do the sequencing, we end up with much shorter lengths, so about usually 50 to maybe 300 or so of these letters in order. But there's a lot of them. There's anywhere between 10, 350 million of these short sequences. And so we have to take those and try and do something interesting with them. And the top bit, this is now what we call it a reference genome. This is not something we've generated for this experiment. It's a common resource, which has been generated through a massive public effort. And we take our custom experiment, these short sequence reads, and we try and align them, just line them up against and see where they go, where they came from. One of the projects that we've worked on a lot at SciLifeLab, headed up by Maxine in the audience here, is called SARAC. And this is an analysis pipeline. And what the primary thing we do here is we look at where these reads that we've generated from a sample line up against a reference genome. And importantly, we look to see if there are any parts of those reads which differ from the underlying reference genome. These are called variants, DNA variants. So where this person is different from the general kind of consensus. Of course, it's not quite as simple as I made it sound. So you have to think about whether it's just kind of background noise or whether we see it across lots of different reads and whether we have good evidence for these variants and 100 other things that I won't go into detail about. But this is really cool. Once you've got to this kind of this final decision about this thing that you might think is important, you can then start to use that for some biological insight. For instance, is this a mutation which leads to some kind of disease? Can we use it to predict some kind of external, like hair color or something else which could be useful? And so then, even after the latter stages of analysis, finding out whether this is interesting or not is a huge task. So typically for SARAC, for this analysis pipeline, we'll take in samples, maybe 2 or 3 different samples from a single patient, cancer patient. We'll take in like a sample of normal cells, which are healthy. We can take in cells from a cancer, and maybe if that patient's been sampled and then suffered a relapse, we can take in cells from that relapse. Or we can compare those variants, because some of them will just be normal, common variants which are not disease-causing. But by comparing these different samples, we can figure out which ones are new, and then maybe we can have some insight into how that cancer has come about and perhaps how best to treat it, and actually have some real impact. And these are the kinds of ways that we can use genomics to actually affect modern healthcare, personalised medicine. Okay, so back to the technical slides. We have this reference genome, which I said we're lining up these reads. And without this, we can't do any of this analysis. It's not just one genome, of course. There's lots of different species. I've talked about humans here, but maybe you want to look at chickens or dogs or cows. There's lots of different species out there. Each species then has multiple different versions of a genome. When you sequence a genome, there's not just one of it. You can keep sequencing more people and getting better and better versions. So just like software, you can keep improving that resource. So there's multiple versions of each genome. There's different tools that you can use to use the reference, and each one of these needs its own kind of set of indexes. So then there's multiple different index types for each version. And of course, this ends up with a lot of files. And so in order to make this a bit more manageable in the compute environment, we've put together, with the help of the guys at AWS, a little resource called AWS iGenomes. Basically, it's a very simple thing. A company called Illumina, who make the sequencing machines I was talking about before, gathered much of this public data into a single place. But just as we heard earlier, it was on, it still is on an FTP server as massive tar files, which weigh a huge amount of download time and contain lots of things that you don't need. And so years ago, we downloaded this to our local server, which took about a month, and it sat there and we've used it ever since. And that's great for us, but it's a big hassle. So what we did with AWS, our genomes, is I just stuck it on S3. At its simplest, this is just a glorified FTP server where now we can pull specific files that we want to our analysis, which can be running anywhere. But of course, as Joe described, the best thing about this is that we can now start to bring the data and the compute to these reference files, and they're just instantly available without any download steps, which saves a lot of time and therefore money. This is the what was it, Rhoda? The open data portal where you can find AWS genomes. And there's also a website for it, which you can go and find. And this just basically helps you get into that data. It's lots of files, so this is kind of just a little helper tool to try and get you to the ones which you're interested in. You can see here I've selected human. I've said that the data should come from a specific source, a specific version, and I want to use a specific tool. We've got some S3 commands here just to quickly copy and paste into the terminal. And there's also a bash script to kind of automate this on the command line if you're already in that environment. And perhaps what's most useful is that these URLs, these S3 objects, identifiers are static and stable. So we can refer to these in code that we generate as well. So if you're interested, go and just Google it or find the URL. And you're welcome to use it as much as you'd like. Okay. Pipelines. Analysis pipelines. When we run a genomics analysis, we have a bunch of requirements for how we do that analysis. Typically, analysis pipelines have multiple steps in them. Maybe 10 steps for a simple pipeline, maybe 50 for a complex pipeline, and they can have complex logic within that. So we don't want to just do that in a bash script, and we certainly don't want to do it manually. We need those steps to be parallelizable. If you put 10 samples in, you want all 10 samples to run at the same time. And if possible, you can split up that analysis to run as quickly as possible. It needs to be reliable, it needs to be portable, so that other people can replicate your analysis, and the results need to come out the same every time. This is not as trivial as it sounds, and it's taken the bioinformatics field, as we call ourselves, many years to grappling with this. And now there's lots of noise about how difficult it is to reproduce published results because of these phenomena. To get around this, we're using a tool to build analysis pipelines called Nextflow, which is developed in Barcelona by a team at the CRG. And what Nextflow does, it's what's called a DSL, domain-specific language. It's based on Groovy, which is based on Java. And you write these little scripts which just basically describe what types of analysis steps you want to run, in what order, and how they connect. So you have inputs, you have outputs, and then you have basically a bash script or some kind of script that you want to run. This isn't specific to biology and genetics. It's been developed in that field, and it's become widely used in that field, but you can use this to run any pipeline you like. And I know there are people doing, for instance, meteorological research using this, and other domains as well. And the beauty of Nextflow is that it abstracts what you're interested in about doing your analysis away from the bare bones of the back end. So it's the same kind of undifferentiated heavy lifting that Jed mentioned. So, for instance, this script that I just showed you is a very, very short, basic script, of course, and I can run it with a simple Nextflow command. By default, that would just run it on my laptop, and it would assume that the software is installed, which is kind of old school analysis, where I have to do much of that heavy lifting of installing the software and figuring out how it works. But if I want to, I can just use an easy flag and tell Nextflow to use different software packaging managers. So now I can tell it to use Docker instead, and then I don't have to worry about the software installation for this pipeline anymore. Everything is automatically fetched for me and wrapped up nicely. Equally, if I have a nice analysis cluster that I can use somewhere, and I can tell Nextflow to make use of that hardware in a more intelligent way. And Nextflow supports lots of different types of hardware. So AWS is there, but also my old-fashioned academic compute cluster, which uses Slurm, is also supported, Kubernetes, and like a whole host of other other hardware stacks. Nextflow was actually built with cloud computing and AWS in mind from the very start, and as such, it has really, really nice native support for it. These different steps in that pipeline script that I showed are nicely kind of wrapped up individual tasks. Each one we can define what the CPU resources are, what the memory requirements are, and what the maximum amount of time it should take. And of course, we have a Docker container for each one of those different steps. And this makes it absolutely ideal for AWS Batch, which is one of the newer services brought out on AWS, where we can just submit the pipeline. Nextflow handles all the back-end stuff for us, and a separate instances created for every single task with just the software we need and just the exact requirements that we need, which makes this very, very cost-effective and basically infinitely parallelizable. So now we have a cluster which we only pay for just the minutes and exactly the resources that we need, and it scales as wide as we possibly can go with all of our samples, so our pipelines run faster and cheaper. It also works beautifully with S3, so there's native support for object storage, and so typically we will set these pipelines up with a couple of buckets. We can have an intermediate one where, with a data retention policy, where those intermediate files are automatically deleted after a couple of weeks, but they're still there if we want to resume the pipeline before that. And then we have a more stable results bucket where we can set up automatic data archiving to cold storage. And this works just really, really nicely out of the box. So to give you an example of a real-world pipeline like this is Sarek again, and you can see there are loads of different steps within this pipeline, and they kind of come together to take the raw data through to something which is actionable, hopefully currently by researchers, but now we're talking about how that could be actionable by clinicians. That's not the only pipeline we've written, so more recently in this past year we've started to develop lots of different analysis pipelines for different types of data. And to save everyone from reinventing the same wheel, now we have a system where we can build these pipelines which run on any computational hardware using any kind of software packaging. We can actually start to collaborate on these analysis code across multiple different institutes, which has been something which has been fiendishly difficult to do in the past. So we have this initiative which we call NF Core, and we have lots of different research labs from across the world now, all contributing and using the same analysis pipelines, which is pretty unheard of. Again, there's a website which you can check out, and if you're into software development, it tells you that all of our pipelines adhere to a set of standards which you will hopefully be familiar with, things like automated code testing, software management, documentation. So if you use one of these pipelines and it works for you, you can be pretty sure that all the others will also work. And there's a good community to help you out if not. We have about, what does this say, 16 pipelines at the moment. It's growing very quickly. And so there's lots of different types of analysis you can do here. Everything from eagers for ancient DNA, so you can put in your Neanderthal genomes and analyze them. We have DNA sequencing, like I talked about earlier, and you can do immune profiling, all kinds of different things with these pipelines. Because all of these pipelines are built with Nextflow, they all have Docker support, they all use the AWS iGenomes references that I mentioned. It means they're perfectly set up both for AWS, but also key for us is that they also work with traditional compute infrastructure. And this is really important because in an academic setting, we haven't been able to use cloud computing until now, until there's actually a cloud computing resource within Sweden. And so we've been building these pipelines for maybe 3 years on our existing compute infrastructure. But we know that when the day comes, we're able just to flick a switch using new config profile, and our code will work on the cloud transparently. And none of that work will have been wasted. We won't have to reinvent the wheel. And so this portability across different infrastructures, including this first-class support for AWS, is really like the key selling point of this code. Okay, with that I'd like to wrap up. There's lots of people being involved in this work. People at the NGI, and also people involved in NF Core, and the guy who's behind Nextflow, Paolo, they're all key to making all this come together. There's various different website URLs which you can check out for more information. All of our code that we develop at NGI is on GitHub from the word go, so you can find all of our open source code. And we have this opensource.SciLifeLab.se, which lists all of the different packages that we've released, which is a kind of a one-stop shop to find information. All of the cloud testing that we've done, as well as the IG names, has been made possible by the AWS Cloud Credits for Research program, so we're really grateful for that. It's been a game changer for us. This wouldn't have happened otherwise. If you're interested, check us out on Twitter. We've got a brand new Twitter account there. Tells you what kind of stuff we're doing. And I'll be a little bit cheeky as well and just follow up with one final slide to say that we're looking for new data scientists. So even if you don't have a background in biology, but you find this kind of stuff inspiring and you want to do work which really kind of makes a difference, then come and talk to us.